Transcription is a process where DNA is copied into a new strand of RNA. The -35 region determines the rate of transcription and it assists in the binding of RNA polymerase to the promoter region. The -10 box is a DNA sequence that indicates where the other DNA sequences can be decoded, and it also determines where transcription starts.
The +1 region is the initiation point where transcription first starts.
RNA polymerase is an enzyme that synthesizes the DNA strand by copying a DNA sequence into RNA. If the -10 box was deleted in a gene that codes for metabolizing sugar, RNA polymerase would not be able to bind to it, so transcription would not be able to complete. The RNA polymerase would then bind to the +1 start site and the strand of DNA would start to unwind. The holoenzyme is another helper in initiation that contains subunits and helps the correct initiation to occur in the promoter region. In this diagram, transcription is occurring from left to right, so I have made the bottom strand 3' to 5', which will be my template strand, so that RNA can move from 5' to 3'. The sigma factor in the promoter region is a protein that is used during initiation. It allows the binding of RNA polymerase to the promoters. Elongation is a part of transcription where the RNA strand is synthesized. The RNA strand is complementary to the DNA template strand, which is where the RNA polymerase moves down the DNA. RNA is synthesized with the help of rNTP, which stores chemical energy and is a building block for RNA synthesis. RNA is transcribed from the 5' to 3' end, and since I labeled the bottom strand as 3' to 5', it will be the template strand, and the top will be the coding strand, which has the same sequence that RNA has. Elongation begins when polymerase releases the sigma, and the RNA polymerase moves down the DNA template strand, which synthesizes the RNA from 5' to 3'. During elongation, new nucleotides get added to the RNA strand, making it longer, and the phosphodiester bonds bind the new nucleotides together in the RNA chain. Termination occurs when RNA polymerase receives a signal from a terminator to stop transcribing. There are two types of termination, row-dependent and row-independent. In row-dependent termination, there is a binding site for a row factor protein. When the row factor binds, it will follow the RNA chain up to the RNA polymerase and will eventually pull the RNA chain away from the DNA template strand, which releases the RNA and causes termination. In row-independent termination, RNA reaches the end of the gene that's being transcribed and it contains a lot of cytosine and guanine. The RNA chain binds back on itself, which is called a hairpin. The end of the RNA-DNA binding contains adenine and uracil pairs. This interaction is not strong, so the RNA polymerase detaches and the RNA chain is released. After transcription is completed, RNA will travel to the ribosome where it is received and translated.
We can think of transcription like a restaurant. When you go to a restaurant, the host or hostess tells you where to sit and then give you a menu so you can start talking and looking at the menu, similar to how the promoter tells RNA polymerase where to go on the DNA strand and to start transcribing during initiation. Next, your waiter or waitress comes by and takes your order, which adds onto the list of meals the kitchen staff has to cook. This is similar to how in elongation, new nucleotides are added to the RNA strand and the phosphodiester bonds have to do more work. The waiter or waitress then comes back and gives you your check, which is a signal that the meal has ended. In termination, RNA polymerase gets a signal from the terminator that it is time to stop.